Okay, so so this is part two of the lecture on viruses. Okay, and we just finished on part one, talking a little bit about human immunodeficiency virus, or HIV. Again, the virus that does cause AIDS, acquired immune deficiency syndrome. All right, so you went through the HHMI click and learn, and you would have learned a little bit about the replication cycle of the HIV virus. And over here on this particular side, we are looking at how HIV does become incorporated into your T cells in your immune system, and it only gets incorporated into the T cells of your immune system. So I'm going to look. We're going to look at an animation right here about how that process does occur. It's about an 8-minute animation. And so, like I said, this video is about it's only 4 minutes long, not 8 minutes. My bad. Typical retrovirus, meaning that it has an outer envelope, and in the center it has two copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine coreceptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the two the cell membrane and the viral membrane together. And what ultimately happens is fusion of those two membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein shown here in green and red that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. Here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point, coalescing with other envelope proteins that have been produced, you have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced. that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
All right, so that was the end of that little video right there, looking at how HIV does infect your cells. Okay, all right, so let's move on a little bit. So, what can viruses do? Well, basically, they can do several different things. They can damage or kill cells by releasing hydrolytic enzymes. So, that's one thing they can do. And again, that's going to basically lead to apoptosis, programmed cell death. They can produce toxins that lead to diseases. And sometimes, they can their damage of viruses can easily be repaired, such as, like I say in here, your respiratory epithelium after a cold, but it can also cause permanent damage, like in your nerve cells we see in polio. This is the smallpox was a scourge that has been ravaging human populations for thousands of years. 1977, when I was 15 years old, was the last case of smallpox recorded in the world. It's the only viral disease that we have eliminated from the world. HPV. HPV is a very common viral disorder. It leads to warts. Probably one of the most common virus disorders that we have, and it can be sexually transmitted in terms of genital warts and things of that nature. And we now have a vaccine for it. If you are a female, when you get a pap smear, that's basically what they're looking for. Are abnormal cell growths, because these can cause cancer. Okay, and now we have a vaccine against HPV, which is suggested that any child or adult, any child over the age of 12 years old should be getting this HPV vaccine, male and female, because it is so widespread and it can lead to cancer. And a vaccine that can prevent cancer seems like a pretty good deal to me. All right, I'm going to kind of skip over some of this stuff. The only thing I do want to say is that vaccines can act in lots of different ways. And when you look at the HIV video, you saw that there were many different steps in an HIV infection. Well, many of the drugs that we produce to help basically prevent the symptoms of HIV or destruction of your T cells play a role in one of those steps. They prevent protease from cleaving the viral proteins. They prevent integrase from getting the viral DNA into your host cell's DNA, and so on and so on. Okay? Anyways, we're starting to see all these dangerous emergent viruses. Just a few years ago, it was Ebola. Currently, it is the coronavirus, with close to 500,000 deaths in the United States, and estimates to be anywhere from 600,000 to a million deaths just in the United States by the time this pandemic is over. So it's huge. Okay, the Spanish flu in 1918 led to an estimated 60 million deaths worldwide. Hopefully, the current pandemic will not reach those numbers, but we're not out of the closet yet. All right. So again, the emergence of these new viral diseases due to these three processes: mutation, which happens, as you saw, very easily with COVID, or end HIV, the spread of existing viruses from one species to another. Okay, rabies is a great example. Ebola is another example. The reservoir for rabies can be in raccoons, it can be in dogs, it can be in cats, it can be in bats, it can be in skunks and all sorts of animals. For Ebola, we think the main reservoir is in bats, also as it is in SARS-CoV-2. And again, due to worldwide travel, remember any disease in this world is only 24 hours away from Trussville, Alabama. Because of this rapid transmission and rapid travel, any disease can travel from these small, isolated populations to the worldwide stage, so to speak, in a rapid manner. Um, So this is kind of I like this kind of little slide here saying that again, 3/4 of new human diseases have originated in other animals. So this zoonotic transmission is an example again of as we interact more with the wild, how that can spread disease, and as we start to destroy rainforests and things of that nature, or we start to move into areas where these animals were, we start to expose ourselves more to these viruses. Um, you can read this slide, but I'm going to pretty much skip over it. And same thing with this one. I just want to talk a little bit about SARS-CoV-2. These are phylogenetic trees, and we'll be hitting this up strongly in our evolution unit, which is our next unit. But we know that SARS-CoV-2 originated in bat populations. Some of the genetic markers indicate that the SARS-CoV-2 virus has passed through pangolin populations, and a pangolin is like a scaly anteater if you've ever seen one. And these are examples of phylogenetic trees down here. Where you see these branching points, these are called nodes. This is where a mutation occurred. And so we got 1 mutation, 2 mutations, 3 mutations, 4 mutations, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 different mutations shown here. Each one of these mutations leads to a different clade, and this would be one clade of viruses that are genetically similar to each other. So a couple of different hypotheses how this may have happened. We'll look at some other ones a little bit later. So this is kind of a cool, I want you to click on this animation button down here in the bottom that you will see on your slides at home, and that'll show you a great animation of how SARS-CoV-2 has spread around the world, where it was prevalent in the beginning and where it's prevalent now. So look through that animation, it's pretty cool. This is another phylogenetic tree looking just at the mutations that has occurred in the SARS-CoV-2 virus, with the original virus over here in December of 2019, and we see that there are some major mutations, and those mutations led to basically 2 major clades. We have this clade over here, and we have this lower clade on the bottom. Well, this clade over here then underwent several more mutations, as we saw in the lower clade also. And this one only goes to March of 2020. Oops. Sorry about that. It only goes to March of 2020. This is a much larger model now, showing many more mutations. And as you well know, we now have the British variant, which is more contagious, now has been confirmed in Jefferson County, which means that there's probably hundreds of cases of that new, more contagious variant here in Jefferson County. Another mutation, one that arose in South Africa, was recently, as of yesterday, was discovered in South Carolina. A little worrying issue with the South African variant is that it is not really responsive to the vaccines, meaning that we're going to have to retool some of the vaccines in order to basically take on this new variant if it gets a strong foothold in the United States. And again, this is why we study evolutionary biology. This is why understanding phylogenetics and natural selection is so important, because it allows us to look at these changes and make predictions and prepare for changes in these viruses and things of this nature that can directly harm us. So I'm going to stop there for today. This is the end of the virus lectures. Next week, we'll be going into both eukaryotic and prokaryotic gene regulation. So we'll hit up those next week. All right. Hope you got it.